Max Srivastava, and welcome to our spring symposium. Many of you I see many friends in the audience and are familiar with us here at Gladstone, but I also see many new faces. So let me just say we're going to have a great program for you today on stem cells, but I think it's safe to say that the topics that we're going to discuss are far beyond stem cells and really touch on the fact that as we sit here today in 2019, the convergence of multiple different technologies and approaches are finally allowing us to begin to really see and feel how we're going to address human disease in a different way in the coming years than we ever have been able to in the past. And so we want to give you an inside view today of how that might happen over the coming years and what kind of steps are leading to that. So before I introduce our topic today, for those of you who are new to Gladstone, let me tell you a little bit about Gladstone and what this symposium has been all about. So we have a President's Council at Gladstone that is many of you are part of, that really are people who've been involved with us or committed to our cause and have been supporting us for many years. And twice a year, we put on, pick a topic of interest to you all and dive deep into that, sharing with you where that field is at the moment. And at Gladstone, we focus on three major diseases of humankind. We don't do everything, but we do basic discovery in heart disease, which remains the #1 killer worldwide, brain diseases like Alzheimer's, Parkinson's, Huntington's, ALS, a number of neurodegenerative diseases that collectively remain a vexing problem where virtually every clinical trial so far has failed to make significant inroads. So it's a major unmet need. It takes long-term commitment, and we're determined to overcome that, and you'll hear about some ways that I think we finally can do that, or can envision doing that. And then we've had a long-term commitment to solving HIV and AIDS and other viruses, and I'm glad to say that in 2019, this is an example of a disease going from being uniformly a death sentence to a chronic disease, completely transformed that disease. And we've turned our attention to other viral diseases and how those viruses interface with the immune system. And how can you leverage that information to engineer the immune system to tackle not only infectious diseases, but inflammatory conditions, cancer, and many other areas where if you trained your immune system, you could use that to your advantage to solve human disease. Now, in all those areas, the pace of science and pace of discovery and translation has markedly accelerated in recent years, and it's done so not by chance. It's done so because technology is advancing at a very rapid rate, and that's what's driving the science. And so at Gladstone, we have recognized that and formalized our effort there by developing an area where we are recruiting talent in developing new technologies to address biomedical problems and using data science to be able to analyze the information that comes from those technologies and using artificial intelligence and machine learning in ways that will give us insight that we could never gain before. And so some of those technologies we've focused on in the last few President's Council symposia, including artificial intelligence last spring, CRISPR gene editing technology in the fall, and some of you are aware that Jennifer Doudna, who discovered gene editing technology, is running a second lab here now at the Gladstone Institutes. And today you'll hear about another technology that is driving this, which is the induced pluripotent stem cell technology that has allowed us to make stem cells from any one of you in this room. And now we can make your stem cells, we can edit them and engineer them with CRISPRs, and we can analyze those using data science in a way that finally allows us to understand human disease. And you'll hear some from one of our speakers, Steve Finkbeiner, about bringing all of that together. And so before I introduce our speaker, I wanted to acknowledge several folks that are here, particularly those who are now leading our President's Council. Will Evers is here, is co-chairing that with Laura Dorman, who's here also, and Fred Dory couldn't make it here, but is our third co-chair. And so they, along with the rest of the President's Council, have become our ambassadors to let people in our community know about the wonderful work going on here. And my only request from all of you is you're going to get some wonderful information today, and then when you leave here, if you're excited about it, tell your friends. And that'll be the most one thing you can do for us. So let me spend just a moment then framing our topic today before I introduce Shinya Yamanaka, who will kick off our comments. So Shinya won the Nobel Prize in 2012, and many of you are aware of this, and it's commonly thought that, envisioned that he won it for the stem cell discovery you're going to hear about. In fact, what he received the Nobel Prize for is the conceptual breakthrough that if one knew enough about every different cell type in our body and how that cell worked, then you could control that cell's fate at will and take any cell in your body and turn it into a different type of cell if you just knew the code that was telling that cell what it was going to be. And so Shinya figured that code out for embryonic stem cells and was able to convert a skin cell into something that behaved just like a human embryonic stem cell, obviated the whole ethical debate around human embryos, but also for the first time allowed us to have access to human cells of brain cells, heart cells, liver cells that didn't require biopsy of your tissue. But when he made that observation, the natural leap forward, next step incarnation of that would be, well, if you could tell a skin cell to become a stem cell by cracking the code, maybe you could turn a skin cell into any cell type you want in the body, not just a stem cell, but maybe directly into a heart cell or a brain cell or a liver cell. And in fact, we've been able to do that now, and investigators here at Gladstone have done it for heart cells in my lab. Sheng Ding's lab and others in the neuro area have done it for various brain cells, as well as liver cells. So I'm going to show you a short video in a moment here that conceptualizes this. So if you see these balls on the top of the hill, imagine them rolling down into a valley, those being individual cell types. It used to be thought that those could never go back up the hill, that ball, and be reset in time. But in fact, Shinya's discovery showed that we could, in fact, introduce just a small combination of genes into those balls, those cells, and push those cells back up the hill so that they are now reset and can go down into any one of these valleys. Okay? So that's the conceptual aspect of this. And what my lab and other labs here have done, if you go to the next video, is not just going back up the hill and then back down the hill, but taking one of those balls, one of those cells that's in a valley already, and it's already determined what kind of cell it'll be, introducing the set of genes that'll tell the cell what to be, and rather than going back up the hill, to be able to sort of jump over a valley, if you will, over a hill, and go into a different valley without having to get reset. But all of this was made possible simply by the conceptual advance that every cell in our body has a key sort of code that only involves 3 or 4 proteins that are enough to tell that cell what it should be. So the only hurdle here is figuring out what that code is. And fortunately now, collectively in the world, we've figured out what that code is for most cells and can now control cells almost at will. And it's opened up a whole set of approaches for regenerative medicine and drug discovery, and that's what we're going to talk about today. So with that, let me introduce Shinya Yamanaka, who had trained here at Gladstone as a postdoctoral fellow. He's one of our alums. Went back to Japan where he was, and he'll tell you a bit about his story. We were fortunate to recruit him back to Gladstone in 2006, 2007. And I'll just tell this story because I think it says a lot about Shinya, and it says a lot about how he thinks of this organization. I had just moved here in 2005 and was building out a program in stem cell biology as well as heart biology. And we had talked on the phone. I had flown to Japan. I flew into Tokyo. Shinya's in Kyoto, where he runs an institute there also. He flew up to Tokyo. We met over a sushi dinner for 2 hours. And by the end of the dinner, he stuck out his hand. We hadn't talked about any numbers, recruitment, and he says, "This is what I want to do." And we shook on it, and that was that. That's how this happened. But it speaks to the trust that one can have between individuals and institutions, and that's what we pride ourselves on here at Gladstone. Shinya's continued to pioneer in our field of stem cell biology. He's served previously as the president of the International Society for Stem Cell Research and goes back and forth between Kyoto and here, but his only laboratory that he runs now is actually here at Gladstone. So, Shinya?
So, thank you very much, Deepak. Good afternoon, everyone. I think I only have this microphone, so I should stick here. So, as you heard from Deepak, I got my postdoc training a long time ago, actually 25 years ago, when I had more hair. So I was a physician before that, but I really wanted to come US, and I applied to as many institutes in US as possible, but I did not hear back from any institutes. But Tom Inerarity at Gladstone Institute, he gave me a positive reply. So we talked over phone. I was in Japan, he was in US. Tom asked me many questions. I forgot most of them. I only remember one question he asked to me. He asked me, "Shinya, do you work on Sunday and Saturday?" And I immediately answered, "Yes, I will." And that was it. He decided to recruit me. So as soon as I joined Gladstone 25 years ago, Tom asked me to convert mouse skin cells into stem cells, which we designated iPS cells. I presented this iPS cell generation for the first time in a meeting in Canada, early 2006. As soon as I finished my talk, a young scientist came to me. I thought he was a postdoc or a graduate student who was trying to ask me a question. But it turned out he was then the director of Gladstone and the future president of Gladstone, who was trying to recruit me. It was Deepak. So we met again, as you heard from Deepak, in Japan, and it took only 2 hours for me to decide to come back to Gladstone. I came back in 2007, and later that year, 2007, we became able to generate human iPS cells. So now we can make stem cells, iPS cells, from each of you. All we need is just a small amount of skin fragment or a tiny amount of blood cells. And the procedure is very simple. In only a few weeks, we can make your own iPS cells from each of you. Once your cells become iPS cells, we can expand iPS cells as much as we want. up to a billion or even more. And then, after expansion, we can convert iPS cells to many types of cells, such as brain cells, heart cells, liver cells, pancreatic cells, or immune cells. So now we can make human cells, many types of human cells, anytime when we want. We are now trying very hard to utilize this technology to help patients. We can use these cells in cell therapy by transplanting like brain cells into patients suffering from heart disease or spinal cord injury. And we can use cells from patients' iPS cells in drug discovery. Importantly, this iPS cell technology also led to yet another important breakthrough known as direct reprogramming, which Deepak just described. So now Deepak can convert scar cells in heart after heart attack back into heart muscle cells without going through iPS cells. So now we really, really want to help patients suffering from intractable diseases with these stem cell technologies. We are still on our way, but we will do whatever it takes to make this happen. So your friendship and your support will be greatly appreciated. So thank you very much for your support.
With that, I'm going to ask Kathy Ivey and Steve Finkbeiner to join us on stage, and we'll have just a chance to have some questions and answers that I'll moderate, and then we'll open up to some questions from the floor. Kathy Ivey is a scientist who trained here and worked at Gladstone for many years, and 2 years ago started working with a startup company called Tenaya Therapeutics in South San Francisco, focused on heart failure, and she'll tell us a little bit more about that. They're using these technologies, reprogramming technologies, to try to address heart disease. And Steve Finkbeiner is a senior investigator here at Gladstone and is doing beautiful work on neurodegenerative diseases, and particularly using human iPS cells from patients with a whole host of neurologic disorders, and as I mentioned, combining CRISPR technology and artificial intelligence to really make breakthroughs. So maybe Kathy and Steve, you each say a word about what you're doing, and then we'll go into some questions.
So as Deepak mentioned, I'm at Tenaya Therapeutics, where we're developing drugs to treat heart failure. And the work that we're doing comes straight out of the findings from our scientific founders here at the Gladstone Institutes, Deepak and other cardiovascular researchers here. And we are working on exactly what was just introduced. So our two approaches at Tenaya for drug discovery are, one, using human iPS cells to make human cardiomyocytes that have mutations that cause diseases of the heart. We can observe those diseases in the cells, characteristics of those diseases in the cells, and then look for drugs, small molecules, or other targets that can reverse those disease characteristics. And we're finding success in that so far. I'm really excited to say, just 2.5 years into it, and progressing some of the work that originated here. On the other side, we're also using these direct reprogramming approaches, exactly what was just described, to transform scars which form in a heart after a heart attack back into working heart muscle to restore function of the heart after injury. And we're taking a gene therapy approach to do this. I'm excited to say that we've progressed it very far since our origination in 2016.
Thanks, Kathy.
And let me add my welcome to Deepak's. It's so great to see what a turnout. Thank you very much for coming. So I'm a director of the Center for Therapeutics and Systems Biology here at Gladstone. And you know, if you want to call it, we celebrated our 190th consecutive failure in clinical trials in Alzheimer's disease last year, if you want to call it a celebration. And you know, I think there's been a lot of hand-wringing about why it's been so difficult to treat brain disorders and move things from our lab out into the clinic successfully. But certainly one of the big questions has been whether differences between the main model we use in science, the mouse, and humans might be a source of some of this failure. And so I couldn't have been more thrilled when Shinya had made his discovery, because I just have a really hard time getting people to sign up for brain biopsies. So having Shinya's technique was really the first time I could ever make a human brain cell in the lab from patients who had a clinical diagnosis. And it's still the case that 90% of patients with Parkinson's, Alzheimer's, ALS have those diseases for reasons we don't understand. And so we can't even answer the question right now, are those one disease, 10 diseases that all look alike? We don't know. And these are really critical questions for being able to understand causes and find therapies. So as Deepak mentioned, we've been working really hard to use these iPS cells. We've amassed one of the largest collections of patient-derived iPS in the world for neurodegenerative and psychiatric disorders. And so we're creating models in the lab that we can use to understand what's causing the disease and then to find therapies. And some of these have been really promising. We've been able, in fact, last year to start a new company around one of the discoveries that emerged from these iPS technologies.
Great, thank you, Steve. I think what you can tell, even just from hearing these brief comments, is that at Gladstone, I think our scientists work with clearly a sense of urgency. We have a large number of physician scientists here as well who take care of patients who are suffering from many of these diseases we've talked about. And it's front of mind all the time that we've got to do everything we can to accelerate the pace at which we make discoveries, because people are dying in the meantime. And so, Shinya, along those lines, I know that you've been very committed to seeing the iPS discoveries get into clinical trials, and have had some success with that. It might be interesting to update the audience on where we stand now with iPS-derived products being actually in a clinical trial and what's on the horizon.
So, as I mentioned in my presentation, there are two major medical applications of this technology. One is cell therapy, also known as regenerative medicine, and the other one is drug discovery. So as for cell therapy, many applications are going on in Japan and also in US. In Japan, almost exclusively iPS cells are being explored, whereas in US, both ES and iPS cells are being...
So embryonic stem cells.
Yeah, embryonic stem cells. So I'm more familiar with the situation in Japan. In Japan, at least two applications are already in clinical trial. One is for eye, age-related macular degeneration. My fellow scientist, Masao Takahashi, she has conducted clinical trial on 5 patients already, 2 years ago already. And the other application is Parkinson's disease. Another fellow of mine, Jun Takahashi, he performed on 1 patient last year, and he's planning to perform 6 more in 2019. So those 2 are already in clinical trials. In addition to that, 4 applications have been approved for clinical trial. One is for heart failure, spinal cord injury, and corneal disease, another eye disease, and platelet transfusion from iPS cells. So those 4 clinical trials are coming in probably this year. And we have other applications, such as type 1 diabetes, joint disorder, like cartilage defect. And also, what is, at least to me, what is very exciting is cancer immunotherapy. So we can rejuvenate and we can expand cancer-attacking T cells and other immune cells from iPS cells. So they are very close to clinical trial as well. As for drug discovery, we have now performing clinical trial for patients suffering from FOP. FOP is an extremely rare disease. We only have like 80 patients in Japan and 1,000 patients worldwide. In those patients, muscles become bones. They suffered from ectopic bone formation in his or her muscles everywhere. So in the end, they cannot move. But we found, by using iPS cells from patients, the cause of this ectopic ossification, and we found one existing drug may be helpful for those patients. So we are testing by clinical trial right now, and many more are coming. We are planning to start one clinical trial for Alzheimer disease and another one for ALS. So it's been 12 years since human iPS cells. So I don't know our pace is fast enough or not. As Deepak mentioned, iPS cells are from patients. So they have like patient's name on each iPS cell line. So we are always under a lot of pressure. But at the same time, by seeing those patients gives us a lot of motivation as well. So it's very unique situation by using patient-derived iPS cells.
I think one thing that we're all so excited about in this area is that unlike most types of medicines that we are used to, that accept the fact that one has disease and you work around that, the stem cell biology and regenerative medicine changes that paradigm completely. It says that we won't accept that we have this disease. We get to the crux of why somebody has disease, and we fix that problem once and for all, and are not just mitigating the consequences of it. And that's what's created so much excitement about at least regenerative medicine, is that we're finally in a position where we don't have to accept the human condition. So, Steve, I know that you have taken some really creative approaches in working nationally and internationally with consortia to collect banks of patient cells, generate them, and then use artificial intelligence approaches to really understand what's going on. Could you say a little bit more about that?
Sure. So just as everyone in the audience is different than everyone else, one of the challenges of working with iPS cells is that each line is a little different than the other one. And so trying to really be able to study these lines and to get information that we can work on and work toward has been a bit of a challenge. And I don't know if there are any cooks in the audience? All right, so a couple. Yeah, good. So, you know, it may take you 3 or 4 hours to make a nice beef bourguignon. It takes sometimes 3 or 6 months to take an iPS cell and turn it into the cell that you want. And it turns out that that very long process can contribute a lot to the heterogeneity that we get when we try to use these cells to answer some of the questions we're trying to answer. So as Deepak mentioned, one of the challenges in this field is really getting useful information from these very valuable cells that we generate from patients, and how can we both get the information we want and maybe even really leverage some of the tools that are available to see new things. So about 4 or 5 years ago, Google came to us. They said we generated enough data to be interesting, which I think is a compliment. So part of the reason we did that is we've built robots that help us to be able to acquire images of these cells on a scope and scale that most academic labs can't do. And that's been very useful for us to be able to really understand the biology that's going on in these cells, because we can study thousands and millions of these cells, so much larger sample sizes than we can normally do. And it was great working with Google because it really gave us an opportunity to work with engineers who can do artificial intelligence and deep learning at the highest level. And it really blew us away. We could see things in images that humans couldn't see. We published a paper last year in Cell to show an example of that. And it had such an impact on my group that now we've really developed a very large effort around artificial intelligence to be able to both see things in the data that we collect that we normally would have overlooked, but I think even more importantly, and this really kind of gets to some of the things we've talked about tonight, to make connections between what we can measure in a dish and what we can measure in a patient, because I think that's one of the most unique features of this tool, is that this comes from a person, a patient, and we have information from them. And as I told you, we've had really lousy luck in the past moving things from the lab to the clinic, but what if we could use these artificial intelligence, deep learning tools to discover connections between the patient and the cell that we can study in the lab that would guide us and help us make better bets when we move things into the clinic so that we can be more successful. And just as you got a sense from both Deepak and Shinya, you know, I see patients or seen patients and really feel this sense of urgency around a lot of these disorders that patients are suffering. And so the better job we can do to accelerate this process and make it more successful overall, the more we're going to have to offer patients.
And while what we do at Gladstone is very basic discovery, trying to understand the underpinnings of human biology and how that goes wrong in disease, as you see the tagline there, science overcoming disease, is what we're here about. And many years ago, particularly after the 2008 financial crisis, it became clear that the landscape had shifted right below us, where if we just kept making great discoveries, and then made the next great discovery and the next, those, unless we were proactive about getting those to the next step to help people, those were going to sit on the shelf. The capital markets just shifted, and we decided we had to be more proactive. And so at Gladstone, the last many years, we have been quite entrepreneurial to try to bridge the gap so that our discoveries can get into the commercial sector, and to the extent that we stay in our sweet spot of discovery but can still facilitate that next step, we've been very active in doing so. And that's meant, in many cases, launching startup companies that might be incubated here at Gladstone and ultimately get venture funding and then branch out on their own. And our scientists continue to stay involved to some degree to help with their scientific knowledge and judgment to push those. And Kathy here represents, as I mentioned earlier, one of those efforts in what's now Tenaya Therapeutics that was housed initially here at Gladstone. But Kathy, I wonder if you could share with the audience what that's been like to be in that setting, coming from the academic setting to that, and how the interface between the two entities has made a difference for the discovery and the translation.
Yeah, well, now having experienced both environments, I can say I love them both, but they're very, very different. And I think that the interface that you referred to is really key to success, certainly on the industry side. And I think we offer something also to the academic group that we interact with. So I would say that the real difference is here at Gladstone, there's such a energy around discovery and an enthusiasm for making advances that change how we think about possibilities, right? But that's not the ideal environment to move things toward the clinic. The environment to move things toward the clinic has to be much actually more linear. We have to have our sights on a specific goal and not be distracted by what could be the next great discovery, the kind of things that happen here at Gladstone every day. So I've learned a lot by being in this environment, and I value the continued interaction with the scientists here from Gladstone who are still working with us very closely. We need their guidance and their thinking on scientific aspects, but then at Tenaya, our scientists are embedded along with clinicians who think about how to transform scientific discoveries into actual therapeutics, people who think about regulatory paths, how to get things approved by the FDA, and then also groups who are thinking about how we manufacture drugs, because how we make therapeutics for academic research purposes is very different than how we make them for putting them into a person. And so we have to have our sights on those differences and making sure that the quality of both is similar.
Great, thank you. So going back to the iPS technology and the transplantation we talked about, when Shinya first made his discovery, it seemed like it would be the ultimate personalized medicine, because we could take a small blood sample from any of you if you needed more heart cells or brain cells, convert those into your stem cells and then your brain cells and transplant them back into you. That way, your body wouldn't reject those cells because they'd be your own. That was the hope that we all had, that it would really personalize your own stem cells. It's turned out that the time it takes to make those cells and the cost, at least currently, is prohibitory to everybody having their own stem cells to be able to use, because you have to get an FDA approval for each one of those. And Shinya went through this and found, I think, maybe it cost over $1 million or several millions of dollars to get a cell line, one cell line, to that stage. So, so Shinya's lab has been working on a solution to that. Shinya, do you want to, if you could say something about how your group is approaching this problem of rejection? If we can't do that, is there some other solution so that you could transplant cells without having to suppress somebody's immune system?
Yes, that's really important from a practical point of view. So once again, my friend, Masayo Takahashi, she conducted the very first iPSC-based treatment for AMD, age-related macular degeneration, 5 years ago already, in 2014. In that case, she used patient-owned iPS cells. And surgery went very well. The patient is still very happy by the outcome of the first transplantation. We were happy to some extent, but we are not so happy in terms of the money we spent and the time we had to use in preparation. So as you mentioned, it took more than $1 million equivalent for just one patient, and it took almost a year to prepare cells from that patient. So it's not practical. There are many millions of AMD patients worldwide. So now, as an alternative, we are making iPS cells not from patients' own cells. Instead, we are making iPS cells from so-called super donors. So super donors are rare, 1 out of like 500 or 1,000. We may have one super donor in this room, maybe 10. So those super donors have very specific immunological types, so that even when their cells are transplanted to somebody else, it won't cause massive immune rejection. It may cause some small immune rejection, but the degree is much lower.
Sort of like an O blood type.
Exactly.
That you might be familiar with, where you can get a donor whose O type can give you blood and you won't reject it.
But other than blood transfusion, our cell transplantation is much more sophisticated and complicated. So each person has a different immunological type, we have more than like 10,000 different immunological types. None of you should have the same immunological type unless we have identical twins in this room. But those super donors are very useful. We have generated iPS cells from 4 super donors in Japan, and those 4 iPS cell lines alone can cover up to 40% of all the Japanese population. Well, Japanese population is less diverse. In the case of US, you need a lot more. In each ethnic group, we calculated we would need 100 lines to cover like 80% of each ethnic group. But it's not 1,000 or tens of thousand lines, 100, something like that. So it's more affordable, but still we need 100 lines or even more. It's a lot of work. To make one line, it still costs almost $1 million US. But thanks to Jennifer and CRISPR technology, we can now make super donor iPS cell lines by ourselves. We don't have to identify those rare super donors. And by modifying their HLA, immunological type, all we need is just a few, like 10 lines would cover most of all the population worldwide, not only for US or for Japan, but worldwide. All we need is less than 10 lines. So it's getting more and more affordable, feasible. And at the same time, we are making iPS technology even faster, even cheaper, more affordable, so that we are expecting in like 5 years, we can make your own iPS cell line with a very affordable price, less than one small car, and within 1 month. So that's our ultimate goal, to make your own iPS cells at the affordable cost and very quickly. So it's still ongoing, but I think we can do it.
Yeah. So, so this is an example of the power of bringing different technologies together. So having Jennifer's lab here and Shinya's lab here with the sort of founders of CRISPR technology and stem cell technology, and how marrying those two can allow efficient alterations, engineering of the genome for the immune system and for many other purposes to study human disease. And it's one of the strengths that we have here at Gladstone, is that we've been able to be at that forefront. So let me open up the questions to the audience. I'm sure many of you have questions here. Sharon?
So as I understand it, one of the issues with cloning...
Oh, and if I could ask you to use the microphone. We are live streaming this and recording it so that the other audience can hear it.
Can you hear me better now?
Yeah.
As I understand it, an issue with cloning is that the cloned creature, like if I wanted to clone my dog, I talked to Deepak about this a few years ago, my old dog, there would be a puppy that would be the result of the cloning, but it would have the age characteristics of the donor dog. So in other words, I wasn't really going to get like a new, younger little puppy. Do you have these issues with the cells? In other words, when you take a skin cell and turn it into a heart cell, does it have the vigor and strength of a baby's heart cell, or does it have the possibly diseased characteristics of the person from whom that stem cell was made?
Actually, it's a very good point. So, for example, now there are 2 ways to make neurons from patients suffering from neurological diseases. One is by going through iPS cells, but the other way is to utilize direct reprogramming. If
By crossing one of those hills.
From like skin cells directly to neurons. If we go through iPS cells, we rejuvenate cells. So even when we make iPS cells from like 80 years old patient, iPS cells, the age of cells is 0, or close to 0. So it's rejuvenation. But if you directly transform like skin cells from 80-year-old patient to neurons, those neurons are still 80 years old. So it turned out it is easier to use direct reprogramming to recapitulate neurological diseases which is affected by age. But if you go through iPS cells, we may be able to observe the process of aging once again. So there are pros and cons on those two projects. Probably, Steve, you should have more.
Yeah, so just like Shinya said, for some of the diseases we study, aging is the #1 risk factor. So trying to understand how that contributes to disease is really important. In addition, as far as we can tell, some of the mechanisms by which your cells age are similar to how environmental exposures may contribute risk to disease. And that's a big unanswered question also. So I mentioned earlier that only in about 10% of cases of Alzheimer's, Parkinson's, ALS can we identify a gene that patient has that accounts for the disease. So we think that there probably are other influences, genetic or environmental, so being able to retain that information from a cell from a patient could be really valuable as well, to be able to study that interaction and to study that mechanism.
Chuck?
Yes. I have two questions, and not being a medical person, please correct me if my English is not proper. Thank you. The first one has to do with converting the HIV virus to a cell because remodeling it and removing the HIV portion, because apparently this virus is very fast in moving through the body, and I just read some things about that, so I'd like to hear a comment on that, number one. And the other one I've read about recently about modifying the LEC, which in the brain, we've talked about the other part of the brain, but the LEC portion of the brain, and working on those brain cells. And there have been some work on mice that's been apparently very successful to help with memory. I just wondered, just comments. Thank you.
Let's see, I see Melanie in the audience, or is Warner or Leo here? Warner? Warner, do you want to comment on the HIV question? Warner is in our director of the Virology and Immunology Institute and is studies HIV.
Thank you, Deepak. So the CRISPR technology in the case of HIV has been a bit disappointing in terms of there have been efforts to try and go in and molecularly remove the virus, but it turns out that the virus can really rapidly evolve and change and make repairs, et cetera. So that approach has not been as effective. We are much more interested in using a nuclease dead version of the CRISPR. So trying to recruit to the site of the virus enzymes that will put that virus into a deep sleep, make it completely inactive and dormant, hopefully permanently dormant. But so it uses the CRISPR backbone, but there's no cleavage, there's no editing of the DNA. It's epigenetic. We're making DNA methylations and histone methylations, et cetera. So that seems to be, that has worked beautifully with various somatic genes.
Thank you, Warner. Pitch?
I have a question that's not scientific, I think it's political.
All right.
It's probably naive, but I have a question that's probably political and perhaps naive. But you mentioned earlier that the FDA would not okay individual stem cell solutions to people's their own stem cell solutions. Well, what is there in the law or in the FDA policy that would not prevent a technique from being used. In other words, a standard technique that was used on everybody. I don't see quite why they wouldn't permit that to be a standard technique similar to what you might have as a patent.
So I think if I could answer that, it's not that the FDA wouldn't approve the use of somebody's personalized iPS line. They would, but they would require that every person's cell line be sequenced in its entirety, evaluated for whether any mutations might have arisen in the process that could be, say, cancer-causing. And so there's a tremendous amount of effort that would have to go into each one of the cell lines being approved for use clinically.
They couldn't approve a technique then.
So I think the technique would be approved. You've experienced this in Japan, Shinya, so maybe you can.
So we are still in the early stage. Those regulatory people, or PMDA here, EMA in Europe, and PMDA in Japan, they are very conservative. So they want us to test each line in animals prior to human clinical trial. It takes very long. But in principle, they are okay with the technology itself. It's just each cell line is different, may be different. So that's what they want to double check. But I'm hoping after like 5 years or 10 years, after 100 patients are treated by this technology, and if we don't see any major side effects, it won't be necessary anymore. So we could apply this technology to many patients without animal testing. But we are still in very early stage. That's why FDA is very conservative, and we should be conservative too, because safety is #1 priority for us.
Some of you may remember in the 90s, there was a lot of excitement about gene therapy. And there was a thought that that could cure so many human diseases. And then there were a few cases where people died in the process of the use of gene therapy, and it completely killed the field for almost 2 decades. There was so the stem cell community has been very careful to not let that history repeat. And so I think even the regulatory agencies certainly have been, but even the scientific community has been rather conservative in moving these things forward to not have any negative outcomes that would just set the field back for many years. So that's part of the concern. I should say that the Japan's regulatory agency has been very progressive, and that's why many of the first trials have been done there. The FDA in the U.S. in the last few years has, I think, had a palpable shift in its risk tolerance. And Scott Gottlieb, who's been the FDA commissioner the last few years, has, I think, done a remarkably good job. And the pace of approvals has increased. And the 21st Century Cures Act, many of you are familiar with it, that Congress passed last year, has tried to make it easier for many of these novel therapies to go forward. So I think there's hope there from a regulatory standpoint as well. Yes.
I was really intrigued by your the therapy that you talked about, creating T cells from iPS cells, and I guess I'm wondering, how do you deal with autoimmunity? So my understanding is, during normal development, T cells are kind of exposed to self, and any T cells that bind to self get killed. But if you're turning like a skin cell into a T cell, how do you deal with, so that it can fight cancer, which is great, but how do you, I guess, prevent it, or how do you know if you put that T cell into a patient if it's going to go and bind to something that it should not?
It's also a very good point. So actually, autologous transplantation, there's no problem, such problem. But when using allografts, we have to consider that aspect. So even making T cells, we should match HLA, immunological type, between donor and recipients. Or, as I mentioned earlier, we can utilize CRISPR technology to get rid of immune-presenting molecules on iPS cells and T cells so that they won't be recognized by donors' immune cells. So at least in animal model, it works. But we need to wait for the result of clinical trial. CAR T therapy is available. It has been approved in this country and will be approved in Japan for leukemia. It's very effective, but it's very expensive, almost $500,000 for each patient. We really want to reduce the cost, 1/10 or even less, by utilizing iPS cell technology.
And one of the interesting features of around the T cells is that we can now make iPS cells from T cells. And when you do that, you make a stem cell that has the same code, if you will, for recognizing self or non-self as that original T cell did. So now you've got a stem cell from that T cell that when you turn it back into a T cell again, it retains that. And I think that's one of the solutions. But Shinya, you raised the point of the expense of this and so many other treatments where, unlike a traditional pill that you might take for 30 years, and a drug company then sells it to you in a pill you take every day for 30 years and they make their money that way, many of these treatments are a one-time treatment, and you're done. So the pharmaceutical that's invested billions of dollars in developing that drug now is going to treat you one time. You're going to pay them one time. And so one of the challenges that many in the community are facing is how do you reasonably price that product? And some of you are familiar with when Gilead a few years ago developed a remarkable, amazing drug that literally cured hepatitis C. Curative. There aren't many cures in our field. Cured it, and they charged $90,000 for it for a 3-month treatment, which is nothing compared to what somebody with hepatitis C would cost our system. Yet they got raked over the coals for that being so expensive. So now you're going to talk about $500,000, $800,000 treatments, and I think we have as a field to figure out how to price that, how to explain that and amortize that maybe over time. But I don't know, has that come up in Japan or others in our audience? We have many experts in our audience from pharma who I know think about this, so I'd love to hear from any of you as well.
Yeah, it's a really important point, the cost of innovation. Like CAR T is very expensive, Opdivo is very expensive, but its effectiveness is remarkable. But in reality, we are going to see more and more these very expensive, innovative treatments in the very near future. So the question is, like here, each country has different insurance system. Are all patients, can all patients access to those innovation, or only those rich patients can access? It's a huge issue. You are right. Many diseases can be treated by just one treatment. So in total, it may be cheaper comparing to like 30 years lifelong treatment. But you have to pay, even though only once, you have to pay that amount of money. And many patients cannot do that. So that's a problem we have to think about. We want to bring our technologies to all the patients, but we may end up bringing our technology only to a small number of patients. So that's what we have to discuss.
Yeah.
It's a huge issue.
Pitch, I know you're very concerned about the ethical and political solutions around a lot of these things, and it's just something we all struggle with. Will?
This is a little different topic. Steve, this might be for you. Several years ago, there was a presentation here around the protein that Dr. Maley was working on for Alzheimer's. What's the status of that research?
Yeah, so the question was about a protein called apolipoprotein E, and I mentioned to you that in most cases we don't understand why people develop Alzheimer's disease, but depending on which flavor of ApoE you happen to have, it's the #1 risk factor for Alzheimer's disease besides aging. And so the particular flavor that raises your risk for Alzheimer's disease the most is ApoE4, and you can think of it a little bit like eye color. You know, we each have a gene that either determines whether we have blue eyes or green eyes or brown eyes, and depending on the variant that you have, you may be at higher or lower risk for Alzheimer's disease. So there's a lot of discussion now about why, what is it about that flavor that confers the risk of Alzheimer's disease. There are a number of ideas about how that might work and whether it's something that we can treat or develop a drug against. And one of the spin-out companies from Gladstone has really been focused on trying to develop therapies that might target that risk factor for Alzheimer's disease. And research here continues on that score of really trying to understand what it is about ApoE that confers risk. There's some thoughts that the protein itself may misfold and form structures that are directly toxic to cells. There's another thought that it may get truncated and affect the sort of energy-producing organelles in cells called mitochondria. Another thought is that it plays a really critical role in the immune or inflammatory system in the brain, cells called microglia. And there's certainly been a lot of evidence from genetics lately in Alzheimer's disease that those cells play a really important role in determining our risk for Alzheimer's disease and for clearing some of the proteins that can misfold in that disorder. So I'd say it's a very active area of research, and because it's so important, I think it's going to be a really exciting direction for the future.
And Yadong Wang's group here and Bob Maley's group have made iPS cells from a number of patients with the ApoE4 mutation and used that to look for drugs that might correct that, some of the problems there, and just reported last year some success with that in identifying a potential therapeutic that could convert the bad form of ApoE better to the good form. And so that's also moving forward. Questions? Yes.
Question for Dr. Finkbeiner. Steve, there's been some really elegant work that's come out of your group recently. And I wanted to follow up on a point you made earlier, and that is specifically a common theme of neurodegenerative diseases tends to be poorly soluble proteins which accumulate over time, gradually gum up the works and cause disease. How do you approach that in the context of stem cell technology, newly created, for example, neurons? How do you recapitulate that model?
Yeah, so thank you for the question. So it is true that one of the remarkable common threads of neurodegenerative diseases is that in almost all of those examples, Alzheimer's, Parkinson's, ALS, Huntington's, if you look at patient brains, you see abnormal deposits of protein either inside cells or outside cells or both. And so it certainly has been a prevailing view in the field that probably plays a really important role, or it's at least a sign of a mismatch that patients may have between the propensity that proteins have to misfold and aggregate and the ability the body has to clear it. And so, as you mentioned, a big focus of our work, or what we discovered as we observed cells, was that there are a couple different clearance pathways in cells, and we discovered some small molecules that stimulate one of those clearance pathways, and they seem to be really helpful for a number of those disorders and for clearing those proteins, at least in our laboratory experiments. And so we're trying to push those forward. It's really interesting in the stem cell area because, as Shinya mentioned, one of the remarkable features is that if you try to determine, so we now have a way that if I took a cell from your body, I could measure a few things called methylation marks on your DNA, and I could tell you within 99% accuracy your age. And so, yeah, it's remarkable. And just like Shinya said, if you reprogram those cells, you erase those marks, and the cell looks like it's a newborn cell. And we've discovered, I would say, that one of the consequences of aging is that some of the systems that cells have for keeping proteins in good shape and for clearing misfolded proteins declines with time. And so what we find is that in stem cells, those systems are really, really good. In fact, it's really difficult to make proteins misfold and deposit in stem cells. So in fact, one of the ways that we've tried to model these cells is sort of directly reprogramming cells to retain those aging marks so that we can make sure that the environment is similar to what the environment might be like in cells from patients where the misfolding occurs, so we can understand that and make sure that whatever we develop that might help boost some of those processes in patients is likely to work in a patient cell and not just a stem cell. But I would say that I think there are huge unexplored opportunities, kind of to Shinya's point, that maybe we can use this technology to also understand better that aging process. And are there ways, we believe that those marks that get placed on cells are things that emerge from pathways that we might even be able to interfere with or modulate. And so maybe there might be a way to even go after some of those mechanisms that are involved with aging and tackle not just neurodegenerative diseases, but other diseases of aging, including diabetes, heart disease, and things like that. So I think there's some really exciting opportunities there as well.
Steve, you've often used the analogy of a flight recorder, I think. Maybe not. Maybe I've used that analogy.
Keep it going, Deepak. Let's see. Hope it's not a 737 MAX flight recorder.
Not that flight recorder. But the notion that when a patient presents, let's say, with Alzheimer's or other diseases, that disease has often been brewing for decades. And so we're only seeing the end stage of that disease process. But the one benefit of resetting the cell and then is that you might be able to actually observe the disease process that might have occurred over a lifetime, but in fast forward and stimulate it and actually catch it early in the stage, would be equivalent to decades earlier in the patient and actually see what went wrong early on. Because when we see the patient ultimately and it's already happened, maybe we're not going to learn from that, how it started. And what you really want to know is how did it start so you can stop it there. And that's, I think, one of the benefits. That's what I meant by the flight recorder.
Thanks. Throw me a bone. Yeah, no, I think it's a really good point. And I just add one more thing, and that is the really good news is that although, we think about these diseases developing, when we're late in life, in fact, you probably have amazing coping mechanisms that exist in your brain that stave off those symptoms for many, many, many years. And so I see that as sort of the glass half full, because I think that as we understand what those mechanisms are, and I think that's where some of these technologies can inform us, that may give us new opportunities to boost just our natural mechanisms for staving off those diseases. Because if we can stave off Alzheimer's for another 10 or 20 years, that's almost as good as a cure.
That's right. Clark?
So, Deepak, it's amazing, astounding things that you're doing and the progress you've made and everything. But we've also hear and understand how much money it costs. So what is happening in terms of funding, for example, the government funding, federal government, NIH type of thing? The state of California, I think, had some biotech fund some years ago. What's happening there, what's happening with other foundations, individual contributions, because obviously there are spectacular things that you can do, but it costs money.
Yeah. No, it's a really good point. And the sad part of that story is that just at a time where our opportunity to understand and address human disease is unparalleled. Just at that time, our funding for biomedical research, at least in the United States, has, in real dollars, been declining. So if you look over the last decade, when all these remarkable things have occurred, the real dollars that are being spent by NIH is down by about 25%. And the opportunity is enormous. And so, institutions like ours, we do disproportionately well in our scientists being able to compete for those funds from NIH. But the real dollars that make a difference actually have been for many in this room, and hopefully others in the future, that allow us to, and our investigators, to take risks and to seize some of these opportunities that are in front of us quickly and make discoveries. So we utilize, it's really been philanthropy in large part that's been able to meet that difference, that need, where the need is. And we use those dollars to recruit new talent, to provide seed capital to take risks and do the really important initial experiments to make discoveries, and to stay at the cutting edge with technology and the equipment that we need. So those are 3 areas where we can't get funding from the NIH, and that's where philanthropy steps in. And the other is our interfacing with the commercial sector. We, as I mentioned at the beginning, we've been more entrepreneurial than I think most. And out of our $80 million annual budget at Gladstone, now every year about $5 to $6 million is coming through our commercial relationships, and that used to be 0 10 years ago. And so between that and philanthropy and our NIH grants, that's our formula for being able to be as aggressive as we can be. But it's critical. Science is expensive, and funding it is a major challenge. And as president, that's one of my biggest challenges, is ensuring that we have the capital to drive the science with the sense of urgency that we know we need to. And thank you for your support in making that happen, and many others in the room. Question here?
So I work with stem cell transplant here in the city, and I have noticed, me and the doctors there at the clinic, that we have a greater results utilizing umbilical stem cells, as well exosomes, even though that we get less umbilical stem cells than adult stem cells from a surgery. My question is, would you utilize umbilical stem cells as a super donor to avoid the rejections that you mentioned before?
Yes, we do. We have been collaborating with Japan Red Cross so that we can utilize cord blood cells. And we now make iPS cells from both cord blood cells and adult blood cells. They look the same, but when we sequenced the entire genome of adult-derived iPS cells and cord blood-derived iPS cells, we see a huge difference. iPS cells derived from adult cells, they have many mutations, whereas iPS cells from cord blood cells, they have much fewer, very small number of mutations. So we are making mutations every day in our blood, whereas cord blood cells, they are very clean. So in that sense, cord blood is a better source.
Thank you. Yes.
Coming back to this idea of rejuvenation, at least in iPS cells, we know that one of the other diseases that's affected by age really precipitously is cancer. And also that one of the concerns about safety of iPS cells in the transplant is that they would cause tumors. But it would seem from what we're saying here is that there's actually a reduced risk because they're rejuvenated. And also that implication is for neurodegenerative diseases, that there would also be a reduced risk, at least as far as the cell autonomous properties of these cells as neurons. So I just wondered if you would like to elaborate on this as as a benefit of going through the reprogramming to iPS cells?
As you said, cancer formation is probably the #1 risk of iPS cells and ES cells transplantation. There are two types of cancers or tumors after transplantation. One is caused by mutations in the genome. But we can avoid that risk because we are sequencing entirely prior to transplantation. If we see any risky change, we won't use that cell line. But there is another risk, which is contamination of undifferentiated stem cells. We don't transplant undifferentiated iPS or ES cells. Only after differentiation, we transplant. We transplant like brain cells, heart cells into patients. But if there are very small number of contaminating undifferentiated cells, they keep proliferating, and after a year or two, we may see tumor formation. So that is probably the most important point we need to avoid. We have been spending a lot of time and money how to prevent that contamination of undifferentiated cells. So one way is to improve our differentiation protocol so that we won't get any undifferentiated cells. The second way is to purify differentiated cells, removing all undifferentiated cells by like cell sorting. And a third way to prevent that kind of formation is to develop a specific chemical that would kill undifferentiated cells, but not differentiated cells. So we have like multiple layers of protocol to prevent such risk. But still, we are not free from such kind of risks. That's why we are very careful in each clinical trial.
Thank you. Kathy, one of the things that in the field we've often thought about is that if many human diseases that we give different names and have different features, the problems might funnel down to some common problems. We think this in the neurodegenerative field where it's just a different neuron that's affected, but they all have the same problem. And so that if you find a drug to one disease, it might apply to many others. Can you comment on what's transpiring in that realm at Tenaya?
Yeah, so at Tenaya, we're focused on developing drugs for a condition called dilated cardiomyopathy, which can arise from mutations in a variety of genes. So there's a whole bunch of different ways that someone can end up having dilated cardiomyopathy. We have now at Tenaya generated a panel of iPS cells that are identical to one another genetically, except that they each carry a different mutation that causes dilated cardiomyopathy in humans. And so this provides a great model for us to, as I said, identify disease characteristics, in addition, look for ways to reverse them. What we have, with these tools in hand, what we have been able to do for the first time is identify a small molecule that could reverse a disease characteristic caused by a specific mutation, and then see if that small molecule is also effective at correcting the disease if it's caused by a completely unrelated mutation. And this was something we had hoped would be true because it really changes the game for drug development. And in fact, that's what we've seen. So we've been able to find small molecules that can correct the disease characteristics in cardiomyocytes in the majority, but not all, of the genetic backgrounds that we've tested. And this allows us an opportunity then to move toward precision medicine or personalized medicine, where if we understand the genetics underlying the cause of an individual's disease, we can then hope to tailor a drug to them that would have some promise of working in their genetic background.
And this was, I think, one of the early demonstrations of this notion that a cell actually doesn't have that many things it can do when you stress it. And so it's possible that for many diseases, there will be a discrete number of responses that a cell has that leads it along a disease pathway. And so if you can figure those out, then you drug those, then you could solve many different diseases. And Steve's work is leading to that as well for neurodegenerative diseases. And so I think it's my great hope that that sort of approach will simplify the problem for so many human diseases, and we'll be able to find some drugs that affect many things. I think Steve uses the term common threads along many different diseases. Great. Well, thank you so much for your attention today. I hope this has been an interesting program. I'll leave you with the thought and the vision for where we're headed as a field. So I don't think it's unreasonable to look forward in the next few years, next 5 to 10 years, to a day where every child, when they're born, has their genome sequenced. Right now, we can sequence the coding part of their genome for only $250. That's probably not that different than getting your blood count done in terms of cost. So it's not inconceivable that everybody, when they're born, will have their genome sequenced, will know all their genetic variants. We'll be able to utilize gene editing technologies to manipulate those codes. We'll be able to use stem cell technologies to be able to understand what that genetic variant, what the consequences are, and maybe even in some cases correct that. And all that is now not just pie in the sky. It's going to take some time. It's not around the corner. But it's something that is tangible now that will alter the way that we address human disease. And so that's where the field is headed. That's what those are the types of things that we're trying to do here, and doing that in great collaboration with many others, including our colleagues across the street at UCSF and much beyond. Science, fortunately, works as a web where we interact deeply with one another, and that's something that we value here at Gladstone quite a bit. So we have some wine outside and some food for reception, so please join us there. And as I mentioned earlier, I hope you've enjoyed the program, and my only ask for you is to go spread the word. So thank you very much.